Hello, my name's Dr. Zoe Waller and I'm an Associate Professor in Drug Discovery in the School of Pharmacy at UCL. Welcome to my little animation video for Soapbox Science. I work on DNA, the molecule of life. DNA is an information molecule and provides the genetic code for all forms of life on Earth. For example, our bodies are made up of many different types of cells, such as fat cells, nerve cells, and muscle cells. Within each cell is a nucleus, and this is where our genetic information is held in the form of DNA. DNA is made up of 4 building blocks, which we call bases: A, T, C, and G. And these letters are otherwise known as adenine, thymine, cytosine, and guanine. Our genetic code is the sequence of these bases, and the organisation of these letters determines whether we are human, a mouse, a banana, and each type of organism has a different sized genome. So bacteria have the smallest genomes, from 160,000 letters long. And believe it or not, plants have the largest genomes, with up to 149 billion letters long. This is much longer than the human genetic code, which is by comparison much smaller at only 3 billion letters long. But those 3 billion base pairs each fit into a cell in a space just 6 microns across. For reference, human hair is about 100 microns across. So there's a lot of packaging to get that DNA into each cell. DNA codes for the instructions to make an organism, but to be used, a working copy needs to be made. That working copy is called RNA, and the RNA working copy is used to make the proteins that are needed. The DNA are the instructions to make something, and we can think of it like a precious book, one that we would like to keep safe. In this case, we're going to look at a recipe book. To use this book, we would like to take a photocopy of the recipe, a working copy to use. And we would then use that working copy to make the recipe, and this time it would be for cupcakes. The shape of DNA was originally discovered to be a double helix and shaped a little bit like a twisted ladder. But we've found since that DNA can adopt many different types of structures, and these are what my research is focused on. For example, DNA sequences that contain a lot of the base cytosine can fold into special shapes called I motifs. DNA sequences that contain a lot of the base G can fold into special shapes called G quadruplexes. We know that reading the DNA involves enzymes and proteins interacting with the DNA. If we change the shape of the DNA, it will affect the way the DNA is read. If we stabilise these special shapes and structures, we can potentially switch genes on or off. And this is what my research is focused on. We can change the shape of DNA using drug-like molecules, and these could be developed into medicines for genetic diseases. Changing the shape of DNA in soil bacteria might help us stop them producing greenhouse gases. We can also change the shape of DNA by altering local conditions such as pH. So if you take a single strand of DNA, it can act almost a bit like a piece of string. But if we add acid, the DNA can fold up very quickly. If the DNA is folded, we can change the pH to alkaline and it will unfold again. So we can go from folded to unfolded, and this could be used like a switch on or off. We can also use other metals to change the shape of DNA, such as silver. This can also be used to fold the DNA into special shapes. We can also do the same thing with copper, and these types of conditions can be useful for making small machines from DNA, a field of research called DNA nanotechnology. So DNA is actually more than the molecule of life. This little shapeshifter keeps me on my toes in my research, and it's why I love what I do. Thank you for joining me for this video about the changing shape of DNA for Soapbox Science. I hope you can join us for questions. Bye for now.